Okay, so this lecture begins the first of four, I think it's 4 or 5 lectures on virology. Now, virology, the science of virology may seem pretty overwhelming compared to what you've already learned in bacteriology about bacteria, parasites, and so on. The problems is that viruses are very difficult to understand, very difficult to study experimentally. And compared to bacteria, where we don't have an all-encompassing therapeutic drug like antibiotics, even with the advances in medical science over the past 100 years, we still have no really effective means of dealing with these organisms. As an example, the current COVID-19 pandemic, even though we've learned how to transplant tissues, transfuse blood, make artificial limbs, and so on, the best thing we have against this virus is to practice quarantine and face masks that was adopted 1,000 years ago for other viruses like smallpox and so on. And this shows how difficult it is to deal with something that can replicate so quickly, but not only that, can change its structure. Because the only effective means we have of dealing with these organisms is by boosting our immune response to target these organisms, mainly using vaccines. But again, with the COVID-19, it seems like we only have protective antibodies at most up to 6 months. Why? We don't know, because this is very unprecedented. We haven't seen this, anything like this before with any other viruses. So with that as an introduction, a lot of what I'm going to tell you about in terms of virology might seem complicated, but it's not, because in the West, a lot of people, a lot of kids, even going through school, are aware of viruses, which in this part of the world you're not as exposed to, because talk about current virus epidemics or whatever comes through the media, through newspapers, through conversation with parents, and so on. And you as clinicians, you should always be aware in terms of current events, what is going on in the media. If there is an ongoing epidemic of, say, gastroenteritis in schools, you should expect that in your clinic, you're going to see more and more cases of kids coming in with vomiting and diarrhea, and have some awareness of what is the organism, whether it's a papillomavirus or Coxsackie virus or whatever. So you should have a general understanding of all of these different viruses and what they do. So in this first lecture, I will give you some basic introduction to the physical and biological properties of the virus kingdom, to talk about their size in comparison to these other microbes you've learned about, the structure, that because this is such a diverse group of microorganisms, they vary quite significantly in terms of their composition. And then because of their composition, that affects the way they replicate. So first of all, in terms of size, we measure virus particles in nanometer scale, which means 10 to the minus 9. And that is at least 1,000 times smaller than bacteria, which are measured in in the micrometer scale, 10 to the minus 6. And then going to 10 times greater than that are eukaryotic cells, which are around 10 to 20 microns. So cells in our bodies are 10,000 nanometers. Bacteria are 1,000 nanometers, so 10 times smaller. And viruses are 1,000 times smaller. The thing about viruses is that there's always been this constant debate about whether you consider viruses as living organisms or not. The thing is that these viruses are totally dependent on a live viable cell to be able to use the enzymes for its replication. But then viruses having such high mutability are able to evolve. So do we consider that a living organism? The viruses either contain DNA or RNA, which determines how their replication strategy works. There's one significant family which it was a major discovery when it was first found, has the ability to go backwards, starting off as an RNA virus, going back into a DNA genome, and then going back to RNA again. That's why they're called retroviruses. These viruses rely on normal receptors on cells to get into the cells to infect them. Many of these receptors are normal functionally physiological part of the cell. So for instance, the COVID-19 uses the angiotensin converting enzyme inhibitor. This enzyme is involved with regulating blood pressure. It's present in neural cells, present in the respiratory tract, the GI tract, and that's why it seems to cause so much pathology in all of these different systems. The HIV virus, which causes AIDS, targets the CD4 antigen on T helper cells. By killing off all the T helper cells means that people with AIDS have no functional immune system. The virus which causes common colds, in fact, uses the adhesion molecule ICAM1 as a receptor. So basically, these microorganisms, by targeting normal physiological processes, can affect the function of the body. But not only that, much of the pathology, the symptomatic effects of infection, aren't so much due to the virus, but the immune system attacking that virus itself. So all of the symptoms you feel during a flu infection, the high temperature, the fever, the tiredness, all of that is due to the inflammatory cytokines which have been made to combat the infection. So by the time you feel symptoms after being infected, the infection is essentially passed away. All of those symptoms that you're experiencing are actually due to your immune response. This is a scale of all different objects in life. So this is an atom which is 0.1 nanometer. Macromolecules like proteins or lipids are around 1 to 10 nanometers. A virus is around 100 nanometers, between 10 and 100 nanometers. Bacteria, as I said, are 1,000 times bigger, so we go up, jump into the 10 to the minus 3 or micrometer scale. I mean 10 to the minus 6. And then eukaryotic cells are 10 times bigger, around 10 microns. Because of the size of, of viruses, unlike bacteria, which you can use a normal light microscope, as you've used before in the micro lab practicals. For viruses, because of their size, we can only visualize them with a specialized instrument called an electron microscope. So unlike a light microscope, which uses photons to shine onto the object, with the size of with the small size of the viruses, we can only visualize them with electrons. So these are higher, smaller, high energy particles, which are used to bombard, going through this vacuum tube, to bombard a specially prepared virus preparation. These viruses are then coated with a high contrast substance like carbon or heavy metals, such that when these high energy electron beams hit the prepared virus sample, they bounce off, and that reflection is collected on an X-ray plate or an imager, so you can get an image of the virus particle, a negative image. The virus makeup is pretty simple. So think of them as a series of boxes. So the outer box is a protein shell called a capsid. And this is made up of subunits like bricks in a building called capsomeres. Again, also made up of proteins. These are able to self-assemble. So imagine having a pile of small magnetic beads. Once you put them together, they can all fuse towards each other. The same with capsomeres. This is the way they work. They adopt the lowest energy in order to assemble. So they don't need any active enzymatic process to cause them to come together. The whole thing about a virus is that it is the perfect example of creating something with the least energy. Within this outer core or shell or capsid, some viruses have another inner core called a nucleocapsid, which covers the genome of the virus. And this is also made up of protein. And then within this nucleocapsid, or within the genome of the virus, you can also have additional proteins packaged with this tightly bound genome, which are proteins associated with various enzymatic functions in terms of genome replication, in terms of hijacking the cell's synthetic machinery for its own purpose. So you can think of these like virulence factors. With regard to these retroviruses I talked about, these viruses contain enzymes which allow the virus DNA to insert itself into the host cell DNA. And this is the same with another group of DNA viruses called herpes viruses. Once these viruses have inserted themselves into the host cell DNA, it means that you essentially inherit these viruses, because these viruses are part of your genome and they can control when they are expressed so they can hide from the immune system. This is why, have you ever heard of herpes cold sores? Some people, particularly in teenagers through kissing, acquire this herpes virus, which grows up the sensory ganglions in your mouth. And there they can shut themselves down and hide themselves from the immune system. Under hormonal stimulation, sunlight, eating milk products, the virus can flare up again, grow back down the ganglia where they cause fever blisters on your mouth. And this can happen throughout your lifetime. So you get this flare of acute blistering around the lips that may last for about a couple of days to a couple of weeks, finish, and then it goes back up again. So then viruses can either be naked or they can have a lipid envelope. And the generally conventional wisdom would suggest that naked viruses, in other words, that have a pure hard protein shell or capsid, are more stable. And this is true to some extent in that most of the, or all of the viruses that infect the GI tract, such as poliovirus, Coxsackie virus, are naked particles. So they can withstand the low pHs of the stomach, the bile acids, and so on, and then still be viable when they're excreted from the body in the feces. Lipid envelope viruses, on the other hand, because lipids are sensitive to detergents, to drying out, to UV light, and because viruses that are enveloped, all the receptors are embedded within this lipid envelope, you would think then that envelope viruses are a lot more sensitive to being outside of the body in the environment, because, well, this is one of the reasons why, for instance, like rabies virus or the current COVID-19 virus, which is an envelope virus, that the easiest way to kill the virus is by using soap, detergents. Because of the lipid, as soon as you use something like a detergent, you're going to break up that lipid and prevent all the receptors from functioning. But nevertheless, with the current COVID pandemic, where they've looked a lot more closely at the survivability or viability of the COVID-19 outside of the body, it still appears that even with the lipid envelope, the virus is still potentially infectious for at least 1 or 2 days. So this here, which is an example of a herpes virus, this is the lipid envelope, and this is the protein shell or the capsid. And between the envelope and the capsid, you have this fuzzy mass of protein here, which is called a matrix. This is, just think of it like cotton wool, of providing structure to this lipid envelope. Think of it like a plastic bag around the capsid shell. But the important thing to remember is that the outermost surface of the virus, whether it's non-enveloped or enveloped, is where all the receptors are. And these receptors is what the virus uses to get into the cell, and these receptors are the targets you want to block if you want to make a vaccine against these viruses. As I said, viruses can be all different shapes and sizes. One of the most common structures is the self-assembled capsid structure called an icosahedron, meaning, it's from Greek, meaning 12 different faces. Some viruses can have so many of these capsomeres assembled within them that they look more circular, which is what the coronavirus looks like. And then some viruses can look like tubes, like the virus that causes the hemorrhagic fever, Ebola virus. And just to repeat, remember that viruses can either be naked or they can have an envelope. With regards to the gene, the genetic material in the viruses, viruses are generally either made up of RNA or DNA, with the exception of these retroviruses, which are RNA viruses which can go back to DNA. Whether it's RNA or DNA, some viruses have single-stranded DNA or RNA. A lot have double-stranded. Whether it is single-stranded or double-stranded determines how that virus replicates, which I'll tell you about in the next lecture. Some of these viruses, the genomes can be structured in various ways. So some viruses the genome can be as a circle, like a plasmid that you learn with bacteria. Some viruses have genomes which are segmented, so they're made up of 6 different strands, 6 to 8 different strands, like chromosomes. And this is significant. This is particularly in the case of influenza virus, because if you get infected with 2 different strains of influenza virus, you can get mixing and matching of the different strands as the progeny develop, which creates a completely different new virus. And this is why the WHO so closely monitors avian or new strains of virus that appear every year in the world. Because if one influenza virus that normally infects, say, birds, then manages to infect a pig or a cow, it can acquire genes from the influenza viruses that are already in those animals to mix up within to create a completely new strain. So depending on the nature of the receptor of the virus will determine what type of cells, what type of animals, what type of organs the virus can infect. And this is what we call tropism. So based on that, so many viruses are limited to particular organs. So, for instance, the hepatitis viruses predominantly infect the liver. The COVID-19, we now know, can infect the lungs, the GI tract, the heart, the brain. So many of these viruses are also multi-system viruses. And then there's another particular population of viruses which are associated more in tropical diseases, which we call arboviruses, because they rely on vectors to be spread, just like you learned in parasitology. So these are the viruses like the Ebola virus, the MERS virus, which infected the Middle East 5-6 years ago, which rely on a vector. That vector with many of these viruses are bats. Through the feces that they pass out, you breathe in, you acquire these viruses, or you eat these animals. With regard to replication, this is relatively simple. We divide them into a number of steps. First, the virus needs to recognize the receptor on the host target cell, so it can bind to that surface receptor on the cell and then get into the cytoplasm. Once it enters the cytoplasm, that whole capsid structure will dissolve to release the genetic information of the virus. And within that genetic information, we'll have codes for hijacking the host cell's replicative machinery to replicate its own genome, to create or translate its own proteins. And so the virus then increases its nucleic acid and also creates protein components to make up a new virus particles. These can self-assemble, and then the virus can either get out of the cell by budding, if it's an envelope virus, or it can cause the cell to die by necrosis and then just burst out of the cell. We divide the phases of replication based on our ability to observe the replication process under the electron microscope. So in the initial phase, when the virus infects or sticks to the surface of the cells, we can see this under the electron microscope because we can see virus particles stuck onto the surface membrane of cells. Once it gets into the cell and starts to uncoat, we generally can't see anything because this is where those capsids have broken up and the DNA or RNA has been released. And so the synthesis or the replication of these nucleic acids are being done. So essentially you can't see any particles. You just have proteins or nucleic acids. So this is what's called the eclipse phase. After 1 or 2 days, then with the accumulation of new virus DNA or RNA and all of these protein being made, and they start assembling into new virus particles, you can then see that within the cytoplasm. And so this is what's called the productive phase, because this is where new baby viruses are being assembled and accumulated. So if you break the different processes down more closely, so in the beginning, during the attachment phase, as I said, the virus needs a receptor which is specific for particular surface proteins on the cell. This is one way of the virus getting in. Once the two, if this is particularly in the case of envelope viruses, because remember the envelope is made up of lipid, the cell membrane is made up of lipid. So when these two lipids get together, they fuse, and this causes the opening up of this whole capsid structure to release the contents of the virus into the nucleus, into the cytoplasm. Another way that viruses can get in, particularly if they don't have an envelope, is to be phagocytosed. But instead of being phagocytosed into a lysosome, they're taken in through this process of pinocytosis, just like a normal cell would take up molecules. And so they bypass the lysosome and they get into the cytoplasm in these vacuoles where they can just break out and then unpackage to start replication. This process is called viropexis. So once the capsid gets broken down to release the viral genome, it can then start its program of replication by hijacking the cell's replicative enzymes to make more DNA or RNA, to take its polymerases to make messenger RNA, to use the host cell's ribosomes to make viral protein. And generally, if the virus is a DNA virus, it will replicate exclusively within the nucleus of the cell. If the virus is an RNA virus, it will replicate within the cytoplasm, because there the virus needs to provide a lot of its own enzymes. Influenza virus is uncommon in that even though it's an RNA virus, once it uncoats, once it gets into the cytoplasm, it can migrate into the nucleus of the cell to then use a lot of the cell's machinery. So, as I said, because whether the virus is DNA or RNA will determine what mechanism or strategy of replication the virus uses. If it's a DNA virus, it will replicate just like a cell does, going from DNA to messenger RNA and then creating more DNA strands, right? But the common feature of all of these replication strategies, whether it's DNA or RNA, to make more strands of DNA or RNA requires that you have a double-stranded template. Do you understand why? This just comes from basic molecular biology. So if you have, all right, so all of us are diploid, so all eukaryotic cells are diploid, right? So we have a plus strand and a minus strand that make up our DNA. Now consider that to make RNA out of that, what is the template for making the RNA? Sorry? One of the strands, right? But by convention, consider that the messenger RNA is what we call a plus strand. That is the code whereby you translate from that plus strand message your amino acids to make up your protein. So that means that the information to make that plus strand RNA must come from a minus strand DNA. So in other words, you start off with your double-stranded DNA, you use that minus strand, the nucleotide code, ATGC, whatever, to read off a plus strand. So it has to be complementary. And the same goes if you're going to replicate your DNA. If you are a single-stranded DNA virus, in order to replicate more DNA, you need to make a complementary minus strand to provide the information to make up the opposite strand. This will become clearer when I talk about the next lecture. But the other thing to consider also is that with RNA viruses, we have to think of the RNA as not only genomic RNA, but also as messenger RNA, which is easy for us to understand with DNA viruses because we have our DNA, which is the genome, and the RNA, which is the message which reads off our proteins. But RNA viruses, the RNA is performing two functions, one as a genome for new viruses, but the other as messenger RNA. So the enzyme which creates new RNA molecules during these RNA virus replications, we call these replicases. But if that RNA is being made for messenger RNA, that involves a polymerase, an RNA-dependent RNA polymerase. So the same protein might have two different functions, one to create more genomic RNA or one to create messenger RNA. There's minor differences between those two molecules. Another thing to consider is that, as I said, because viruses are the most efficient way to generate an organism with a minimum of genetic information, then they have ways of being most efficient in creating different proteins, different amino acids, with a minimum of genetic information. And so with RNA viruses, they can be what are called polycistronic, where during replication, they will create a long messenger RNA molecule. But this messenger RNA is composed of small packets of messenger RNA information. So this is why we call it polycistronic, where a cistron is a cassette of information. So in other words, during transcription of the genomic RNA into this messenger RNA, you got this long messenger RNA molecule, which then gets transcribed into individual proteins. Another way we use this is that some of these RNA viruses can make a long protein molecule. So they make this long messenger RNA, which then gets translated into this large protein molecule, which then gets chopped up into individual protein components. So this this efficiency of use can lie either at the level of the gene or at the protein, the translated protein. And then another mechanism of reusing genetic information is to use one sequence of nucleic acid, which contains start sites for reading individual messenger RNAs. So for instance, with the hepatitis B virus, which is a circular DNA, right? So this represents the DNA genome of the virus. When the virus transcribes into messenger RNA, one start site occurs here. So this is the one messenger RNA. Another start site is here. This is another messenger RNA. Another one is here. This is another messenger RNA. But this one here can get cleaved just like that into 3 different proteins. So don't worry too much. I won't ask you about these sort of details. But this is just to give you some awareness of the complexities of virus replication. All right. And then during the time course of replication, we group the different molecules of the virus based on when they appear. So the first instance, once the virus enters the cell and releases its components to start firing up the replication, the first proteins which are made are called immediate early proteins. These are the proteins involved in hijacking the cell's machinery to unwind or expose the viral genome for replication or transcription and so on. And as this process goes on and you get more and more new viral proteins being made, then these are what are called the late proteins, where the new capsid components assemble to create new progeny viruses. The new genomes are packaged within these capsids, and then other proteins might cause the cell to die to release these new components. So this is why we have immediate, early, and late. And during this late phase, these proteins might be modified in terms of glycosylation. They might acquire the host cell envelope in the case of envelope viruses, and then released from the cell. So this is just an example of one of these processes. So here we get first the entry of the virus into the cell. The virus particle opens up, releases the genome. The genome moves into the nucleus and then uses the cell's machinery to replicate to create more virus DNA. This DNA codes for proteins, which are then assembled and then released from the cell. And this is a cartoon to describe this. So this is the initial infection stage. The virus is binding onto these receptors on the cell. Can then fuse, get into the cytoplasm. This is a case of viropexis, where it's getting into these so-called Catherine pits. This then causes the capsid to open up and release the genetic material. These migrate through nuclear pores into the nucleus. And there they hijack the cell's polymerases to create more copies of the virus DNA, which then gets transcribed into messenger RNA and translated in the cytoplasm to make more virus protein. Here, these new proteins are modified, glycosylated in the Golgi apparatus. Then they get back into the nucleus to assemble these virus particles, which then bud out of the cell to infect more cells. So, as I said, this course on virology might seem overwhelming, but basically for assessment purposes, all you'll be required to know or remember are the names of common viruses, like what is a poliovirus, herpes virus, viruses which commonly feature in our daily lives. You need to know something about those viruses. Are they made up of RNA or DNA? Are they single-stranded or double-stranded? Then also, do they have an envelope or not? And then know something about what kind of diseases do these viruses cause. So we know that poliovirus can cause paralysis, or it can cause failure of the lungs to breathe. How do they get into the body? They get in through the GI tract and so on. So we'll take a 10-minute break, and then we'll start again at 11. Okay?